My name is Shona Kerr. I'm at the MRC Human Genetics Unit at the University of Edinburgh. And today I'm going to speak to you about genomic medicine in isolated Scottish populations. First of all, I'd like to thank DNA Genotek for providing me with this opportunity to share my work and for helping to make this presentation possible. So in today's talk, I'm going to begin by introducing our VIKING Genes research cohorts. I'm going to describe the recruitment methods for 8,000 people with ancestry from the Northern Isles of Scotland, that's Orkney and Shetland. I'm going to tell you a bit about our progress towards return of actionable genetic results to the research volunteers, and I'm going to touch on underrepresented populations in genomics research. This is quite a busy slide, but I just wanted to make the point that population cohorts kind of fit within a circle, a bigger picture of how we might understand the interplay between genes and environment that together contribute to human health and disease. So the population cohorts I'm going to speak about are from the Northern Isles of Scotland. We'll also hear a lot in this conference about the UK Biobank, and I'll be mentioning some of that data myself. In our population cohorts, we're able to make use of the high kinship to do family studies, and that allows some interesting study designs. Of course, a research cohort in genomics requires samples which can be biobanked and have a whole range of uses, including the generation of new phenotypes, including multi-omics phenotypes. Samples can also be used towards functional follow-up of genetic findings, which in turn have been made from analysis of collected DNA and used for things like genome-wide association studies. So our VIKING genes, I've given a map here of Northern Europe with the red blobs indicating Orkney and Shetland. As you can see, they're actually nearer to Norway than to London. So it's perhaps not surprising that people from Orkney and Shetland have the highest degree of Norse blood in the British Isles and Ireland. This has been known for a while, but we really nailed it down in a recent paper in PNAS. So we have three cohorts making up VIKING genes, one from Orkney, one from Shetland, and one from both island groups. First of all, I'll tell you about the original VIKING cohorts. This was clinic-based recruitment for approximately a decade, ending in 2015. And that work resulted in 4,000 participants living in Orkney or Shetland. And in these cohorts, consent was taken for feedback of the phenotype that was collected at the clinic visit, but not for genetic results. These cohorts over time have supported hundreds of research papers, many with other cohorts, and the blood that was collected in the clinics has been converted into plasma, serum, RNA and DNA, allowing multi-omics. So at the beginning, the DNA samples were genotyped, then more recently we were able to get some whole genome sequencing, and finally we've had all the exomes sequenced. We've got some summaries of our recent papers on the website if you're interested. So research using the increasingly detailed genetic data shows that there's a different pool of rare gene variants in these populations compared to cosmopolitan populations. In this case, we compared the variant load to an Edinburgh population, and that's published in the paper I've pasted in there. And some of these rare variants are medically relevant. Some of them are completely, have no function. And we did a project looking at an actionable variant in the KCNH2 gene, which increases risk of long QT syndrome. And that's published in scientific reports. For this talk, I'm going to define an actionable finding as a gene change that's linked to a condition or disease that can be prevented or improved by treatment. And I may also call an actionable variant an AV. So what we did was to analyse 5 actionable variant carriers that we found in our Shetland cohort. And we were able to show by haplotype analysis that all 5 of these people had a distant relationship deriving from a common ancestor. But in the modern era, these 5 volunteers fell into 2 families. And the 2 families, A and B, were not recorded in the pedigree or in the genealogical records that are available in these populations. And what this means is that the family B that we identified would not be identifiable by cascade testing in a clinical setting. That's where an index case of a genetic variant carrier, pathogenic variant carrier, is used to cascade out into their family. But of course, if the 2 families don't join, then that's not possible. So here we were able to return the results to the research participants via their GPs, and that was because the people show a phenotype, prolonged QTC interval on an ECG trace. So we were able to do that work even though we didn't have consent for return of genetic results, and that was because of the consent that we had. And although we were able to do that work before we had exome sequence, it's important, vital to have sequence data to find these actionable variants that are rare and not visible just through genotype analysis. So we're now busy focusing on analysis of just over 4,000 exomes derived by Regeneron. And I wanted to mention an actionable BRCA1 variant that we think is a founder mutation, because the participants are all descended from the Isle of Westray in Orkney. We didn't discover this variant. It's been known to our colleagues in NHS Grampian Clinical Genetics for many years. And I've pasted in the ClinVar record for this variant, which shows it's pathogenic and increases risk of breast and ovarian cancer. So this particular actionable variant is not found in the GNOMAD database, the online database of exomes and whole genomes. We also didn't find it in our 2,000 exomes from Shetland, but there it is in Orkney, 20 instances. And we also found it in the UK Biobank, 4 instances. And one of our 20 carriers in Orkney has been diagnosed with ovarian cancer. We're able to find that out through our record linkage to the electronic health record. That's the routine NHS data, in this case, the cancer registry. And similarly, of the 3 female carriers in the UK Biobank, one of these has been diagnosed with breast cancer, again, as shown by the electronic health record data. So what we have here is a variant which has a much higher frequency in Orkney compared to the UK Biobank. That's likely due to genetic drift. And we aim to offer return of these results to the ORCIDES participants. We think that this sort of work demonstrates the value of our VIKING Genes research cohorts in more fully describing the burden of rare clinically actionable variants in these populations. And it should support better planning and delivery of genetic services by the NHS. While all this was going on, we'd been funded to extend our cohorts by another 4,000 new volunteers by launching the VIKING 2 study, which is an online study. So here there are no clinics. And instead, we take consent online and participants fill in a questionnaire online. And then we use so-called spit and post saliva collection to obtain our DNA samples. Eligibility for this cohort is to have 2 or more grandparents from Orkney or Shetland. And again, we're taking consent for EHR linkage, which over all 3 Viking Genes cohorts, about 8,000 people. And this ability to follow the health trajectory of the volunteers over time adds an element of longitudinalization to these cohorts. Another feature of Viking 2 is that right from the start, we requested consent for return of selected actionable genetic results. And another relatively unusual feature for a population cohort is that we've recruited worldwide. And I'll say more about both these points in a moment. I just wanted to point out the time course of our recruitment as measured by questionnaire completion, which is the point that triggers us to send a sample kit. So the study recruitment launched in January 2020. We got a great initial response, as you can see by the curve. And it was already beginning to level off a bit by March 2020, when, of course, the UK lockdown began and the team had to start working from home. We were quite fortunate. If we'd been running a clinic-based study, that would have been the end. We would just have had to stop that. Because it's online, we were able to keep going, keep in contact with our volunteers and work effectively from home. It did have an impact on recruitment numbers, but once lockdown ended, we've been able to get new people in, and we're not far off now reaching our 4,000 target. So as I mentioned, Viking 2 is a worldwide study. These maps, each little dot is a participant, and you can see we've got lots of people in the Northern Isles, and nearly half of the participants. The rest of Scotland, particularly down the east coast, we have about 35%, rest of the UK 10%, and rest of the world just under the 10%. So I just wanted to illustrate the worldwide recruitment that we were able to do by showing this world map, and it illustrates that the historic emigration routes of people from Orkney, for example, going to work in the Hudson Bay Company, is still evidenced today by our participants across Canada, and also the emigration of people from Shetland to New Zealand and the east coast of Australia. So the current population in the Northern Isles is approximately 43,000, but there are people, and therefore their genes, dispersed all throughout the world. I just wanted to say a bit more about the spit and post sampling, because it's a technique that really opens up genetic research to all sorts of populations that previously couldn't have been reached. We've used the origin method, which is very easy to use, and we found very high compliance. It lets us sample people who might be needle phobic, and the whole study can be done in the comfort of people's homes. And as I mentioned, this was particularly useful during lockdown. Another point is that the volunteers, after they've provided their sample, they put it in the packaging and send it back to Edinburgh. That's postage paid within the UK, but our volunteers outside the UK actually have to pay for returning the postage. So I think that's a great example of how generous and altruistic research volunteers often are. So this has worked really well. We've got over 90% of the samples we've sent out have come back, and it tends to work smoothly. We had the odd glitch. This one is a very sheepish looking dog who took a liking to chewing the packet, and so we had to send a replacement, but of course that's fine. And sometimes, particularly from overseas, the samples take a while to reach the lab. This one has been all over the place, starting in South Africa, and took 13 months to get back to us. However, the sample was completely fine. The DNA is very stable inside the collection tube, so we were glad to see it back. We've got a big information sheet about return of results. It's quite a hard concept to get across to people, but we think we've achieved that. An important point is that we take a number of clauses of consent, but importantly, the decision as to whether or not to get results returned is the optional clause in our consent form. And that respects people's right not to know, which is, I think, really key for any of this type of work. But gratifyingly, just over 98% of the participants so far have said they would indeed like to get actionable results returned to them. And that's very much in line with another body of research, particularly from social scientists, who've surveyed people and asked them what their preferences would be if given the option. And what we're doing is also in line with the GA4GH policy on returning genomic research results. I think this is a really solid, sensible policy, and was published this year, or last year now, and really it acknowledges the potential medical benefits to now the millions of individuals who are participating in genomics research. That ideally we should be looking at ways to return the actionable results to them in a responsible way. And just to stress that our work in this area is in partnership with our colleagues in NHS Grampian, particularly Prof. Zosha Mead-Broidska and Dr. John Dean. I just wanted, so where we got to with this? Well, we're not quite as far on as we wanted to be. In order to return the results in Viking 2, we first have to get analysis of the DNA, and sampling in a pandemic has its challenges. So the upper graph shows the samples, the kits that we sent out, and we had to pause during the first lockdown. And again, over last Christmas, the previous Christmas, we had to pause once again. Luckily, the lab that receives the samples stayed open throughout the pandemic, so the samples were being collected and booked in and stored securely until such times as they could be extracted. And in fact, the lab had to delay the extractions. They've caught up now, and that was because they were prioritising COVID research, in particular the genomic study, which is led from Edinburgh, and which actually provided automated DNA extraction machines. So we've benefited from that for all that we've had to wait. So we're planning for exome sequencing early this year. And we're expecting to get sort of 1 to 2% of our volunteers carrying these actionable variants. But I wouldn't want to leave you with the idea that it's because of the isolate nature of the Northern Isles populations that people carry the actionable variants. It's true that some of them have drifted to much higher frequency than elsewhere, but there's also plenty of variants that don't exist in these populations. And in fact, the population of Orkney in particular is among the highest life expectancy in Scotland. So actionable variants are everywhere. Most of them act dominantly when present in a single copy. I'm not talking here about recessive variants. And by definition, return of actionable variants is not the same as sharing all genetic data with volunteers. So just to bottom that out, yeah, we're not planning to disclose genetic variants predisposing to conditions with no treatment. And it's pretty clear that the gene lists are changing, and new AVs will continue to emerge. Therefore, I think it's quite important that these cohorts plan for long-term engagement with volunteers. And just to illustrate my point that they're in all populations, this is just a snippet from the paper describing the first 50,000 exome sequences in the UK Biobank. And in that cohort, the consent does not allow return of these results to individuals or their healthcare providers, but it is just over 2%. As part of our recruitment in Viking 2, we asked through social media if our volunteers would like to share their stories, which they did, and you can read those on the website. And in fact, one of the people who got in touch was a lady from the family B of our long QT syndrome project, and it turns out she was delighted to have been given this result, which she had no knowledge about beforehand. I'm just going to finish by saying a few words about another study we launched recently called Traveller Genes, looking at people who have 2 or more grandparents who are or were travellers. We've got a focus on Scottish travellers, but we're also interested to receive participants whose ancestors were Welsh Cale, Irish travellers, or Romani. And we've been able to launch this very straightforwardly because we've essentially repurposed the same successful study design as we used in Viking 2. That's online recruitment and spit and post saliva sampling, as illustrated in the schematic here. And this study is looking to understand the genetics, origin and health of people who have traveller ancestry. The study was actually requested by the community, and the public involvement panel is very important in this cohort. As I say, it's a spit and post process. It's a small study, and we're planning to close it in a couple of months. There's really been no genetic research previously on Scottish travellers, and this is an example of an isolate population that's not geographically isolated, but instead culturally isolated. And so we think doing this kind of work in hard to reach people is potentially rather valuable. So we're going to, in terms of next steps, our plans are to finish recruitment. There's a lot of work to analyse that wealth of exome data, both the data we already have and the stuff that'll be coming soon. The team will be doing research into the rare variant function and lots of multi-omics. That's another talk entirely. But we'll also this year be implementing return of results. We estimate about 100 people in Viking Genes, but then it has wider effect across their family members as well. One thing to point out is that we're aware these are research results, so we verify by a different analysis method. We're actually using the very old-fashioned Sanger sequencing, but it still works and is still very inexpensive and straightforward. And the NHS will also, before they do anything to act on the results, will verify, repeat the analysis themselves, and will also offer counselling. So we think the materials and experience that we're gaining here will be of wider value to other cohorts, particularly because as well as taking consent at recruitment, we're also going back to reconsent existing cohorts. And this seems to be in line with international policy and the wishes of participants. So I've described to you one facet of genomic medicine in isolated Scottish populations. I'd be delighted to take your questions. Just to finish up, I'd like to thank the Viking Genes team, which is Rachel Edwards, David Buchanan and myself, led by Professor Jim Wilson, and also the NHS Grampian Clinical Genetics, the Clinical Research Facility Lab in Edinburgh, our colleagues in the MRC Human Genetics Unit, DNA Genotek, and of course the volunteers, without which none of this would be possible. Thank you. Now I'm pleased to be able to answer some questions on the talk you've just heard. I think the first one that's come in is how can you make the data more accessible? So that's something we care a lot about. Genetic and health data are, of course, categories of personal data. So we have to have a managed rather than fully open access process. And there's some links to that on our VIKING Genes website. So I would ask you to have a look at that if you're interested in accessing the data. Quite a philosophical question. What would you like to see change or improve in genomic medicine across the UK? Well, I think this whole conference is already just on day 2 showing some fantastic examples of the really leaps forward in genomic medicine that are happening now. As a proud Scot, I have to say, I think particularly in England, but we're obviously still part of the UK, and much of it's happening in Scotland as well. So I think lots of good signs. I personally think we could do more to work at the interface between research and clinical practice. I think there's a lot of data siloed, and I think we could learn more from each other. Again, I would think there are encouraging signs in that direction. And I think it's crucial in all these big initiatives, these potentially millions of people having their genomes sequenced and analysed. I think it's vital that we engage with the public and involve the public and patients in genomic medicine. Next question. What is the significance of this study? Well, that's a really good question. I've tried to, I think in the talk, make the point that it's not all about size of cohorts. I mean, we have these remarkable cohorts in the UK and also across the world, such as the UK Biobank, which is just a fantastic resource. But there are gaps, and I think cohorts and projects like VIKING Genes and Traveller Genes help to fill those gaps and do research on these underserved populations, which I think have many benefits for genomic research, not just for the populations, but also for the advancement of knowledge. So we're kind of running down the clock at the moment. I don't think there are any other questions. So once again, I'd like to thank very much the organisers for inviting me to give this talk, and my sponsor, DNA Genotek. And I hope you've found what I have to say of some interest. Thanks.